All right. I'll fix my hair. Important. So good afternoon, everyone. I'm very happy that I can be here together with all of you. I'm a PhD student of BERT. My name is Anne-Catherine, and I'm actually not a physician. I'm from training, I'm a biotechnologist, so I do the work in the lab. So for me, it's very inspiring to see all of you here today. I will present what we are doing in the lab, but in a very simplified way. So if you have further questions, you can always come to me and ask more details or more specific results of what we are doing. So how do we actually start research on genetic diseases? Actually, it all starts with you, with the patients. What we do is, or what my colleagues did before, was a patient identification. So we're looking actually for patients with similar phenotypes, so similar features. And then what we actually try to do is find the specific genetic defect that goes along with it. And that's exactly what our lab did. So how does this actually work? This is a principle of genetic testing. So as you might know or might not know, this is you. And everyone is made up of trillions of cells. So you can not actually see it. In one cell, in a cell, there is a nucleus. And in this nucleus, this is actually where all the genetic material is stored. We can say it like that. Normally, when you see a picture of genetic material, it looks like this. This is a chromosome. And chromosome is just a condensed form of DNA. That, I think, is what everyone knows. So what we do in our lab is we take out the DNA and we study it. It might be difficult to visualize, so I use a figure like this. Imagine your DNA. It's in total 3 billion letters long, and it's actually only composed of 4 letters. We call it A, T, C, and G. And all these letters, they form the DNA, and they form also, we call it genes, and you can look at it as a lot of words written in a book. Your whole genome is a book, and all the words in the book are genes. So what we are looking for is which word exactly in the book is not written right in all the different patients. So imagine it like this. So each patient has a mistake in the same word in the book. We are looking for it. Imagine the word is pear. If you change one letter, it's bear. That's something completely else. Or sometimes you also have not that one letter has changed, but that one letter has disappeared in your genome, and then you have a P. So that makes up for something completely else. So what our lab was able to was identify the exact word that, or the exact gene in which all the ATS patients have one mistake. So we go back to the book to understand it a bit better. In the book, there are about 30,000 words, so 30,000 genes. You know, like certain genes give you hair color, other genes, they work all together, so they can transform the sugar in your food to energy. Other words help you to form new tissue when you have a wound. And the word in which the mistake is in ATS is the SLC2A10 gene, and another name for it when it's a protein is GLUT10. But then there's a problem. For a lot of words in the book, we know what the function is. But actually, we did not know at all what the function was of SLC2A10 when we found out that was the causal gene, the gene with the mutation. And that's actually the start of genetic research. We have to find out what the gene exactly does before we can come up with a treatment or things like that. Of course, the gene was discovered about 10 years ago. Now we're 10 years further and we know, luckily, already a lot more. And what we know now is that the role of SLC2A10 is most likely that it's a transporter of vitamin C in certain cell organelles. So as you could remember the figure of my previous slides, we are all made up of trillions of cells. And in one cell you had the nucleus, where there was all the genetic material. But the cell is composed of many, many other things. You have, for example, the endoplasmic reticulum, and for example, the mitochondria. They are the energy factories of the cell. So all organelles have different functions. And what we know now is that GLUT10 is in the membrane of the mitochondria and the endoplasmatic reticulum. And that's good that we know that, because this helps us to actually pinpoint the exact function of GLUT10. But then, when we know that, we can go one step further. So how do we actually perform research? Of course, we have all of you guys, and we're very grateful that some of you give us a skin biopsy. We can use your skin material to do research on. But of course, we need more. We need a model. We need arteries to look at. If we do certain manipulations, we want to see how our model reacts. So what we do in research is we make animal models. In the news, you hear about a lot of different models. We can use monkeys, or a lot of times mice are used. In our lab, we also use the zebrafish. That's maybe something you've never heard of before. Also, like the fruit fly is used, all different kind of models. And I'm currently studying 2 ATS models. One is a mouse model and one is a zebrafish model. And I will talk about those 2 models a bit more in detail. So the first model is the zebrafish model. I like it a lot. The zebrafish is actually just a normal aquarium fish. But actually the professor that started research on this model, he actually saw that zebrafish has a lot of excellent characteristics to do research on. As you can see, the zebrafish originates from the India and Nepal regions and Bangladesh, where it swims in the Ganges River. But now it swims here in our lab, which you will see later on today. So why do we like to use the zebrafish? It actually has a very fast regeneration time. You can see the life cycle of the zebrafish there. So yeah, the figure might be a bit too small, but at the 1 hour time point, you see the fish is still a tiny, tiny little egg. And actually after 4 days, it already looks like a fish. Not a full-grown fish, but you can see all the organs have formed. So actually this fast generation time, it helps us a lot to perform quick our research. Then you have a lot of other nice features. It has a high genetic similarity with humans. You would not expect that, but actually it is true. It's not to the same extent than with mice, but it is enough to perform trustworthy and reliable research on. Another thing that is very good, because research is expensive, is that it's a small animal, as you will see also in our tour in our zebrafish facility. So it's cheap to house and to feed. Then the figure you see here is a zebrafish embryo of 1 minute, I would say. So what we can actually do is inject certain materials in this embryo, and it immediately influences the development of the embryo. And then, as I said before, it has a super fast development. Here you can see the zebrafish development from 1 cell up to about 24 hours. And you can see the cell division, the cells spreading. Now actually the head and the back of the fish is formed. And it gets all its energy from the yolk, like the big ball in the middle. You can see it here better. For example, this is an experiment that was done. And you can see already after 1 day, you can see clearly that the fish already formed completely, and it stretches further up until 2 days. When we do experiments, for example, injections, like I said before, you can already visualize that there are differences, that we can easily see differences already after 2 days. And this helps us a lot doing experiments. Another very nice thing about zebrafish is that it's transparent during the early development, so no pigmentation has formed yet. And so what we can actually do is put the fish under a microscope, and then we can make special lines, lines that are, for example, fluorescent for the cardiovascular tissue. So the heart is visualized in green and the vasculature in red. And by using this kind of lines, we can see immediately under the microscope, we can look for differences. So all these advantages have led us to making zebrafish models. In our lab, we have made 2 models for now, and we call these knockdown or knockout models. So the difference is you can see it as a speaker or as a sound system. When you hear normal music, it's loud. When you do a knockdown, you just turn the volume down. So the gene is still doing its work, but much less significant. When you make a knockout, you just destroy the gene completely. So the first model we did, we made a knockdown. So we just put the volume a bit down in the gene, and we actually had very nice results with that. So what you could see is we did injections, like you see on the figure here, immediately after the zygote was formed. And then after 1 or 2 days, you can appreciate, like in the left top, you see a wild type. So that's what we call a normal developed fish, so without any treatment. And then all the fish injected with a molecule that made the gene go knockdown. You can see they have a lot of deformities in the cardiovascular tissue. So here we used a special line. Yeah, of course, with the light it's not so good to see, but with using those special fluorescent line, we could visualize immediately all the vascular defects in the zebrafish. So that's good one, because when we can visualize defects similar than seen in ATS patients, we know we have a good model. And if we have a good model, we can use this model, tissues from this model, DNA, proteins from this model, we can use all those molecules to do further study. We don't only look at the model and see what's wrong. We use the model to collect tissue and do further research in the lab, which is maybe a bit too advanced. So I will not go into detail. Unfortunately, this model has also a bit disadvantages. So we see the abnormalities. We can use this model for further study, but the disadvantage is that the knockdown effect only lasts for 4 days. So we can study the fish in early development, but we cannot use the fish to study the disease in later stages. So that's a disadvantage. So what we did then was making a knockout model. So we destroy the gene completely. And we actually do that with a technique. It's very popular right now. You might have heard it in the news, or it's sometimes, really big breakthroughs there in that field, and the technique is called CRISPR-Cas. What the technique actually can do is make a cut in the gene at a very specific place. And what we can do then is just let the cell itself repair the break, but the cell makes mistakes and then you have deletions. But what we can also do now is placing a specific mutation found in patients, placing it in the zebrafish. So that is a much more relevant model. But we're working still on that, so I cannot show actual results from that yet. A second model we use to study ATS is a mouse model. It's also a knockout model, as I explained before. And the reason why we also study a mouse model is because how much I like using the fish for studying the disease. The mouse is way more relevant. It has a heart similar to humans, the cardiovascular or the vascular vasculature is similar to humans, so it's good that we complement our easy and fast and cheap zebrafish model, that we complement it with a more relevant mouse model. Bert previously already investigated a mouse model for ATS. It has missense substitutions, which means that the gene had not a deletion, but it was just one letter in the gene that's changed. And unfortunately, as the title of his paper said, that there was an absence of a phenotype in that mouse model. So what my task was here was to study why that was. So we improved this model. We bought a new mouse, and this mouse actually has a deletion of a big part of SLC2A10, so the gene that we study. Not one letter changed, but a whole part of the gene disappeared. So we thought that was a better option to go for. And then, as I mentioned before, we know that GLUT10 might be a transporter of vitamin C in certain organelles, but actually the mouse can make vitamin C themselves. We humans, we don't. We need to eat oranges, we need to take up vitamin C because otherwise we get scurvy. But mice, they actually are, in that way, they are different from humans. They can make vitamin C themselves. So that might be the case why actually the model that Bert studied actually did not have a phenotype or no abnormalities in the vasculature. So we had a second mouse, and the second mouse had a deletion in the GULO gene. And the GULO gene, that's the function of the gene is that it has a role in vitamin C production. So we were thinking if we knock out both genes, we might get a relevant phenotype, and we can use this mouse for further study. So this is what we call a breeding scheme. We start with the SLC2A10 mouse, we start with the GULO mouse, and then we have several rounds of breeding. And then in the end, we end up with, imagine this as 16 different mice from one litter. So when we have all these mice, we genotype them. That's a bit too technical, but we end up in our study with 4 different mice. The mice completely normal, we call it wild type. Then the mice only mutant for the GULO gene, so the gene with the role in vitamin C. We also use then the mice mutant for SLC2A10. And then we have the red mice here. They are mutant in 2 genes, in both of the genes, SLC2A10 and GULO. And then the 4 mice, of the 4 types of mice, we use them in our study. So the red are the most important to us, and the blue, the gray, and the black are our controls. So what do we actually do then with these mice? First of all, we have to look for a phenotype, so similar features than found in patients. So we first have to determine, is this model a good model to study ATS? So the first thing we do is cardiovascular imaging, because we're looking for aneurysms, stenosis, tortuosity, for things like that. The first thing we did was a big ultrasound study. In this study, that's actually similar to what's happening in patients, although the first step might be a bit different. So first we anesthetize the mice so that they're calm, and then we put them on a warm pad so that they're stable and still. And then with an ultrasound probe, we can actually visualize pretty well the heart, the aorta, the carotids, very similar to what happens in patients. So I put here a small movie. It might be a bit too dark to see, but what you have here is the heart beating of the mouse. Here the aorta comes out of the heart, and what we're looking for is the blood flow from the heart through the aorta, and we're also looking for the diameter, of course, of the aorta. We look at the diameter of the aorta at several points. So this is a figure of how the aorta looks. Here it comes out of the heart. And then we look at different points, like at the root, at the proximal part, the distal part, and just the arch. And what we can actually see, as you remember the colors out of my breeding scheme that I showed in the beginning, the development of this part of the proximal ascending aorta at 6 weeks, 3 months, 6 months, and 9 months. As you can see, the red curve is way below all the other curves. So we actually found a phenotype of these of this double knockout mice, so it lacks the 2 genes, and all the other mice, they are similar. So we were very happy to see that. We had a stenosis phenotype. But of course, the most important thing in arterial tortuosity syndrome is the tortuosity. We would like to see tortuosity of the carotids. And then it would be a very good model to study ATS. We tried that with CT scans, as you might all know, but mice are very tiny. And it's difficult to get a good resolution to see really these tiny, tiny arteries. So we use a different technique, and it's called corrosion casting. This is a really nice technique. It's a bit similar to what is done already hundreds of years ago, but a bit more advanced. What we're looking for is the abdominal aorta. So we look for that in the mouse, and then we put in a catheter. In the catheter, we inject a special solution. It's a solution that polymerizes 15 minutes after you have injected it. So what we actually do is inject the solution. It spreads all over the arteries into the smallest part of the vasculature, and then it becomes a polymer. So it becomes like plastic, it becomes hard. What we then do is dissolve the mouse from around it. It's called maceration, and then after 1 day it looks not so good. But then when you wash it, it comes out beautifully. So this is actually a mouse where every part of the vasculature is replaced by a plastic mold, as you can see it. And then starts my work. What we do is not just look at the mouse, we look at the arteries that are most important for our research. So what I did was I dissected out from each mouse the complete aorta. You might recognize it. Here's the heart, the aorta comes out, and then here you have the carotids coming out of the aorta. Unfortunately, what we could not see in our mouse model were abnormalities in the carotids. We also looked at the arteries, the cerebral arteries, but there we also did not see abnormalities, and we could also look at the retinal arteries. But unfortunately, no differences there. Similar to the figures that Ode showed, we did similar stainings on our mouse tissue than we did on the ATS patient tissue, but there we could also not see abnormalities. So what we have here now is that we have a model with our stenosis, but we don't really have the other typical features like fragmentation of the elastic fibers, or we don't have tortuosity. So another goal for me is to figure out why that is, and maybe if I can induce all these abnormalities there. So what's next? Of course, first we need to show that our animal models have similar cardiovascular features than ATS patients, because we first need proof that they are good models. Then we can use, as I said before, the tissues of these animal models for further study, and then we can elucidate like what is now for sure the exact function of SLC2A10, and then the most important thing to investigate is why do mutations in this gene exactly give the phenotype that we see in patients. And if we have all the links between the gene and the phenotype, if we had laid all this puzzle, we can intervene there. So we can intervene with drugs that we can test on these animal models, but still it will take years to get at this point. But in the end, that's of course what we're looking for. We're looking for drugs that ameliorate your phenotype or might stabilize, for example, dilatations. So that brings me to the end of my talk. I want to thank you for listening, and I don't know if there are any questions.
Thank you.
You're welcome. Yeah. Yeah.
No. Here you go. You know, I can help you clean that mess up. You'll laugh when you see mine.
Which is it? This is the most recent.
Let's see. Yes.
This one?
Mm-hmm.
It's okay if I put it on my desktop?
Yes, you can keep it. How do I change it?
Just by the arrows,
Okay,
forward, backward.
thank you. Hey, Bert.